In the previous video, we discussed the basics of cell signaling, as well as how local and long-distance signaling work. In this video, we will continue further with looking at cell signaling. Specifically, we will go through the 3 stages of cell signaling and the different types of receptors found in the plasma membrane. This video will go in depth and will give you a look at a variety of different aspects of cell signaling and the molecules needed for it. So we begin with the 3 stages of cell signaling, which are 1. Reception, 2. Transduction, and 3. Response. And this was discovered by Earl W. Sunderland, who found this out working with the effects of epinephrine on cells. Now the first stage we will begin with is reception, and reception is when the target cell detects a chemical signal. The signal detected is the signaling molecule released from the cell that sent the message, which is also known as a ligand. Now a ligand is something that specifically binds to another molecule, and this ligand will then bind to a receptor protein and will either promote or inhibit enzyme activity. After this we have transduction, which is when the binding of the signaling molecule changes the receptor protein. For example, it can change the shape of the protein, and this initiates the process of converting the signal to a form that can bring about a specific response from the cell. Sometimes this process can occur in one step, but more often than none, it occurs in a series of steps, known as a signal transduction pathway. The last stage is the response, which occurs when the transduced signal triggers a cellular response. Now this response can lead to many different things. For example, it could lead to the rearrangement of the cytoskeleton, or it can cause the activation of a specific gene in the nucleus. The 3 stages of cell signaling are extremely important for an organism, and must occur properly in order for the organism to function normally. Next, we will talk about some important concepts in regards to the reception process of cell signaling. Usually in the reception stage of cell signaling, the receptor will change shape once the signaling molecule binds to it. This shape change is usually the initial transduction within the signaling. It is also important to note that most signal receptors are plasma membrane proteins. And in regards to the plasma membranes, there are 3 key membrane receptors and a receptor found within the cell. Now the 3 membrane receptors are GProteinCouple receptors, receptor tyrosine kinases, and ion channel receptors. And the receptor found within the cell are intracellular receptors. A G-protein-coupled receptor is a plasma membrane receptor that works with a G protein. And a G protein is a protein that binds GTP, an energy-rich molecule which is similar to ATP. They are also extremely similar in structure, as they all contain 7 alpha helices that span the membrane, which is shown here. On top of that, they are very diverse in their functions throughout the cell, as they are important in embryonic development and also sensory reception, which are completely different spectrums. Interruptions in G protein receptors cause many illnesses, such as pertussis, which is whooping cough, which produces toxins that interfere with G protein functions. And next we will go through how G proteins work. In this diagram, the G protein is connected to the cytoplasmic side of the plasma membrane. Now the membrane acts as a switch, so it can either initially be on or off. If a GTP is connected to it, which stands for guanosine triphosphate, it is on. And if a GDP is connected to it, which stands for guanosine diphosphate, it is off. In this example, it begins off. Here, the G-protein-coupled receptor and the G protein work together with another protein, and in this case and many other cases, it will be an enzyme, shown here. When a signaling molecule binds to the G-protein-coupled receptor, the receptor becomes activated and will change shape, and this allows the G protein to bind to the receptor. GTP then displaces GDP, and the G protein is now activated. Next, the G protein then dissociates from the receptor, moves along the membrane, and eventually reaches an inactive enzyme. When it reaches this inactive enzyme, it binds to it, changing its shape and causing the enzyme to become activated and trigger the cellular response. After the cellular response is completed, the GTP connected to the G protein is hydrolyzed, converting it to GDP. Because of this, the G protein becomes inactive and leaves the enzyme. The enzyme changes to its original shape and the process is available to occur again. So just to go through these steps, guys, again, here's the initial setup. We jump straight to this where the signaling molecule, the ligand, binds to the receptor. This causes the receptor to change shape and allows the G protein to bind to it. GTP then dissociates GDP off of the G protein. And now we're going to jump to the next step. And once that occurs and the G protein binds to it, it's going to go along the membrane and it's going to basically hit into this enzyme. It's going to activate it, and that's going to cause this is going to change shape, and that's going to cause a cellular response. And then finally, the G protein is going to dissociate from the enzyme. The enzyme goes back to its original shape. The GTP is hydrolyzed to GDP, and the extra phosphate is going to leave. Next, we have receptor tyrosine kinases, or RTKs. RTKs belong to a class of plasma membranes that include enzymatic activity. A kinase is an enzyme that catalyzes the transfer of phosphate groups. A tyrosine kinase is an enzyme that catalyzes the transfer of phosphate groups from ATP to the amino acid tyrosine on a substrate protein. So, based on this, a receptor tyrosine kinase is a membrane receptor that attaches phosphates to tyrosines. So let's break this down. So seen in this diagram, in the first step we will look at how an RTK functions. First, we have the receptor tyrosine kinases. In most cases like seen here, these two receptors work together. As of now, however, the receptors are separate entities, or separate monomers. In the second step, binding of signaling molecules to both receptors will cause the two to get closer together, forming a dimer, and this process is known as dimerization. Now, a dimer is simply just two monomers bound together. In the next step, the formation of the dimer causes the tyrosine kinase regions of receptor RTKs to become activated, leading to each tyrosine adding a phosphate from an ATP molecule to the tyrosine seen here. So these 6 phosphate groups come from the 6 ATP that was actually broken down to 6 ADP. And in the final step, after these phosphates bind to the receptor protein becomes activated and leads to relay proteins that recognize the activation to bind to specific tyrosines. This binding causes the proteins to change shape and activate, leading to the activated proteins triggering a transduction pathway, which causes a cellular response. And one thing that's really important to note here, guys, is that the main difference between GPCRs, which is what we just looked at, and RTKs is the ability of RTKs to signal more than one response or pathway at a time. Also, malfunctions in RTKs are associated with many different types of cancers. We also have ion channel receptors. Ion channel receptors are a type of membrane receptor that can act as a gate when the receptor changes shape. The way this receptor works is when a signaling molecule binds as a ligand to the receptor protein. The gate will open or close, allowing or blocking the flow of specific ions through a channel that is located within the receptor. And an example of these specific ions are sodium or calcium. So, let's take a look at this diagram. In the first picture, we have the gated ion channel, which is currently closed and will remain this way until a signaling molecule binds to it. Now, in the next step, when the signaling molecule binds to the receptor, the gate opens, allowing ions to flow through the channel, which will change the concentration gradient along the cell membrane. This change can affect the cell's activity. And lastly, when the signaling molecule dissociates from the receptor, as seen here, the gate closes and no more ions can enter the cell. Some gated ion channels are controlled by electrical signals instead of ligands. Lastly, we have intracellular receptors, which are found in the cytoplasm or nucleus of targeted cells. Very small or hydrophobic chemical messengers can cross the plasma membrane. When they do this, they can activate different receptors. One of these receptors is the intracellular receptor, which will become activated and become a transcription factor, turning on specific genes. Examples of hydrophobic messengers are the steroid and thyroid hormones that are found in animals. In the example seen below, I will use testosterone as the hormone. So first, the hormone passes through the plasma membrane. Testosterone binds to a receptor protein in the cytoplasm, which activates it. So right here, you have the complex, and it forms the hormone-receptor complex. The hormone-receptor complex then enters the nucleus and binds to specific genes. And these specific genes, or specific gene, is represented by this blue dot seen both in the complex and it will be transferred to the mRNA. Now the bound protein acts as a transcription factor, stimulating the transcription of the gene into mRNA, as I just mentioned. And finally, the mRNA is then translated into a specific protein. As always, thanks for watching the video. Please like and subscribe if you enjoyed the content, and if you have any questions, leave them in the comments in regards to this video or any videos you'd like to see in the future.